everyone, welcome back to my channel BioInfoByte. So today we will be covering normalization methods for single-cell RNA-seq and spatial transcriptomic data analysis. The normalization process is used in different analysis to normalize the data. So in this topic, I will be covering what exactly the normalization means and why it is needed, and also the factors we should consider during the normalization step. So in this slide, this is a simple flow chart of NGS data analysis like single cell RNA. You start with the raw data or UMI counts, and then you go for the quality control steps. And then we have the filter data based on the quality of the cells or the parameter we have used to filter out unwanted cells. Then the main steps comes into picture, that is the normalization. And after that we have like dimensional reduction PCA, and then doing the clustering part and visualizing the data or clusters with UMAP or TSNE. So that these steps results are dependent on what kind of normalization you have performed on your data. Normalization is an important process for the downstream analysis and specifically in finding the differentially expressed genes. Why normalization is needed? This is a big question, and there are several points to answer this, why this is needed. So the raw rate counts cannot be directly used to compare the gene expression level between cells. The reason is because they are associated with some of the variation that is technical and uninteresting biological variation. During the sequencing process, there are many technical factors which introduce bias in your data, raw read counts. And these technical factors are like gene length, GC content, sequencing depth, dropout, etc. So these are the factors which introduce bias in your data. And the result of this, the raw data that you have does not reflect the level of biological gene expression accurately. So there are many technical differences between the sequencing platform that will create some noise that makes difficult to understand the actual difference. So the differences we are not sure whether they are the actual biological difference between samples or the differences is because of the noise that is created by these technical factors. So making any interpretation from unnormalized data is unreliable. So there are many QC steps and other methods which are available to filter and reduce the variation, uninteresting biological variations. So for example, while PCR amplification bias is taken care by use of UMI, unique molecular identifiers. But why we use normalization? Because to remove the effects of other technical variations like GC content, gene length, sequencing depth, cell lysis, river transcription efficiency. For these technical factors and their effects needs to be removed. That's why we have to perform normalization on our data. So it is necessary to make the accurate comparison of the gene expression level between samples to make it possible. So what are the factors that we should consider during normalization? So the main factors which are often considered during normalizations are, so first one is the sequencing depth. So in this example, you can see like each gene expression is doubled in sample A compared with the sample B. And this is not because the gene in this sample is showing high expression because of the sequencing depth. Like this, these genes are sequenced doubles times as compared with the sample B. That's why they have more number of reads. The next one is gene length. So in this case, you can see gene X and gene Y have similar level of expression, but the number of reads map to gene X and gene Y. So in this you can see like gene X has more number of reads compared with the gene Y. So this is because the gene length of gene X is greater than or longer than gene Y. The third one is RNA composition. So it is also important for accounting RNA composition to get the accurate comparison of expression level between samples. So in this example, you can see like this is the sample A reads and this is the sample B reads, and here the expression level for three gene X, Y, Z and D genes. Same for the sample B, expression level for gene X, gene Y, gene Z. If we are going to divide each samples by the total number of counts to normalize the data, the counts basically skewed by the D genes because in the samples D is in the dominating part, which take up most of the counts for sample A. But in the sample B, this will not be the same result. Like if we are dividing with the total count, the expression level will be different compared with the sample A. So those genes, for example, gene X and gene Y and gene Z, if we are dividing these genes with the total count, so their expression level will be less. But in this case, sample B, when you are dividing gene X, gene Y, gene Z with the total count they have, the expression level will be higher. But that is not the scenario because of the dominating part of D genes. So that's why normalization is needed. So the normalization process in Surat is the important process in the workflow of single cell and spatial transcriptomics pipeline. So for simplicity, I am dividing normalization into two steps, scaling and transformation. So Surat basically apply a global scaling normalizations method that we call log normalize. So this is a definition that I have taken from the Surat pipeline itself. It normalizes the feature expression measurement for each cell by the total expression and then it multiply by a scale factor. So in the Surat, the default one is 10K, and then we do the log transform this results and then we have the normalized data. So let's see with an example how it works. So log normalization in Surat works in two steps. So first step is multiplying with a scaling factor. So for example, in this scenario, example, we have like two cell types, cell type A and cell type B, and we are checking for the gene expression of A and B. So for example, see without transformation, the analysis will be dominated by the highly expressed gene. For example, the difference between this gene B is 100 times and this gene A is 9. So this will be dominating by the highly expressed genes. So to perform the normalization, the step one is basically the gene A count in cell 1. So for example, this is cell 1, this is cell 2. So if we have to calculate the normalized value for this particular cell, what we have to do, we have to divide 3. So this will be gene A count in cell 1 will be the value 3. And what will be the total count or total molecules in cell 1? So this will be 103. So we have to divide by 103 and then multiply with the scaling factor, that is 10 to the power 4, that is 10K, which is the default in the Surat pipeline. Then after having this value and we are storing in X, then step 2 is the transformation. Transformation like basically calculating the log 2 value. So log 2 and X plus 1, X the value we have calculated in step 1, and this will gives you the normalized value for this particular cell. And same way we have to calculate for this cell and this cell and this cell, then we have the normalized data. So reasons for doing scaling part is very important. So in this example, you can see like on x-axis, we have the cells and on y-axis, we have the number of transcripts detected for each cell. So number of transcript detected is basically the sum of all the EUMI counts for each gene, for each cell. So the idea of scaling is to multiply the value of each gene EUMI counts by some scaling factor. So after we will have multiplying with a scaling factor, for example, this will have some multiplication scaling factor, this will multiply with the scaling factor. So at the end, all the cells have the same transcript count. So the reason for doing this is because different cell types can have different amount of mRNA. Some cells have more mRNA, some cells have less mRNA. So the transcripts detected depends on how much RNA is available in the cell, correct? and then we are not interested to compare the absolute count. Normalization is divided into two steps, that is scaling and transformation. So scaling is basically multiplying with a scaling factor, and normalization has the second part, that is transformation. So we have different transformation methods. So for simple transformation, we have two methods like log transformation that we have seen in single cell RNA Surath workflow, and then square root transformation method. And the second one is the Pearson residuals, that is basically a negative binomial regression method. And under this, which comes under acid transform. Acid transform normalization method is used in spatial transcriptomics data workflow in Surath.